Okay, hello everyone. I'm David Simoncini. I'm from IRIT, the Computer Science Research Institute of Toulouse. And I've been working at CRCT with a team of IRIT researchers since maybe 1 year and a half. We have our office now in the CRCT next door. And we work on many projects, basically applying AI methods or AI models to health science. So this project I'm going to talk about is on what we did with Dr. Sarah Bertoli and Jean-Baptiste Rieu from the hematology department of the hospital. And this work involves many people. It started last year with some master student from Polytechnic, Ibrahim Didi, and then we followed up this year with another master student from the university, Clément. And now we have 2 postdocs working on that project as well, Ashraf and Philippe, and 2 researchers from IRIT and from the CHU, now Jean-Marc is at the CHU. So first about acute myeloid leukemia. It's a cancer it's a liquid cancer. It's a cancer of the bone marrow cells, and it has many different stages. So basically, it has very bad prognostic. So once the patients are diagnosed with acute myeloid leukemia, the doctors decide for a treatment, and then there are 2 issues: whether you enter remission or you don't. And once you enter remission, you can either be cured or relapse or die in remission. And if you relapse, then you enter this cycle of, well, basically being alive until you die. So we have here in Toulouse, we have access to a very nice database that of clinical and biological data that has been collected for the last 20 years. So we have more than 400 variables in this database. About 1/3 of it, 1/4 is diagnostic variables. And then we have information of treatment and response to the treatment and some news. So because this database was acquired over time, a lot of columns are not very well filled. Some were added in the last years, some were dropped. So we have a lot of sparsity in this database. But if we look only at the diagnostic variables, we have very few correlation between those variables, and it is quite nice because if you want to make prediction from this, then if there is no correlation, it means potentially, well, you can obtain information from all of these variables. So then we have different classes of patients in the database. Well, these 2 classes are just the type of treatment that was given to the patients. So ACT stands for intensive chemotherapy, and AZA, it's azacitidine. So it's a treatment that was given to patients that were too weak to be given ICT. So if you look at the profile, it's patients that are much older. And so we can see many differences in the distributions. Here you have the survival in days on the y-axis and some of the variables in the database on the x-axis. So what we did with that, I mean, one of the things we did with that is try to predict overall survival of the patients. at different times. So because the patients were given different treatments, we separated the database into these classes. So we make predictions for one particular class, so one particular treatment. So our workflow looks like that. We first process the data. What we did in data processing is basically trying to reduce the sparsity of the database. So we dropped a few columns, and some columns were merged together in consultation with the doctors to make sure that we don't mess up with the data. And then we applied some AI models. And alternatively, here we applied one algorithm, which is called Boruta, prior to learning the models. And then we have a process of 5-fold cross-validation. So we separate the dataset in 5 ensembles. We train the models on 4, and then we test on the last one. And then we shuffle these ensembles and do this 5 times. And we average all the results. And then, so we have a score, basically the prediction of overall survival. And we use SHAP for model explanation. So the models we used 2 models. We used the XGBoost algorithm, which is some gradient boosting. Basically, it's a decision tree. It's a sum of many small decision trees. So you take, the model tries to find one variable that separates very well the data set into the the classes that we want. So is the patient still alive, or is the patient dead? And then we add up a lot of many small trees like that in order to have refined overall prediction. So it's state-of-the-art algorithm for this kind of data. And then we applied a neural network, so a very, very classic and very, very small neural network that's called multi-layer perceptron, which has very few neurons in it. So 52 here and 20 and 20. And basically, for those of you who don't know, we have the input, which is the data in our database, and we apply transformations to it. And then the transformations will activate or not neurons that will propagate the signal to the next layers. And the goal is to, well, find the series of transformation that will do the correct activations, so that in the end, we have our prediction that is as accurate as possible. So in the end, we have 2 neurons, one for alive, one for dead, and we want to activate the correct one. Then the Boruta algorithm, which I talked about, is used for feature selection. So basically, I will not go into the details of this, but it tells you the predictive power of each variable in the dataset by comparing to a randomized, a shuffled version of it. So if you have less predictive power than a shuffled random variable, then you are eliminated. And so this method is applied iteratively on the dataset until some stop condition. SHAP, which we use for explanation, tells you independently for each variable its impact on the output of the model. So here you have a separation. So if you are on the left, it means we lean towards one class of the output, so say alive. And if we lean on the other side, then it's the other class of the output. And the color corresponds to the value of the feature. Okay, so here it says that, well, it's a random example taken from the internet, but it says that a small age has a lot of impact on the classification into this class, for example. Capital basically has no meaning. So with that, we run our models on the dataset, and here we have many different lines. So the red one is a naive classifier. So over time, here you have the time in months for each patient, and here you have the accuracy of the model. So if you look at after 2 months, almost all the patients in the database are alive. So over 90%. So if we have a naive predictor, I mean, if I want to predict whether my patient is alive or not at that point, I would say, yeah, it's alive. He is alive all the time. And that way, my prediction would be 90% accurate. So that's why we say it's a naive predictor. Basically, it's the majority class in the dataset. So these curves are for the ICT cohort, and it shows that after 19 months, the dataset equilibrates. So there are as many alive as dead patients in the dataset. So the red line is basically our reference. We want to do a lot better than that. And so we compare all the methods to that point here where the dataset equilibrates. So the blue one is the XGBoost. The green one is a very fancy neural network algorithm that was published maybe 3 years ago and specialized on tabular data. And the orange one is a very simple multi-layer perceptron that we tried. And it's always the simple perceptron who has the best performance. We were quite surprised with this. We expected XGBoost to be the best, but well, we did the experiments many times, and the outcome was always the same. So in the end, we have an accuracy of almost 70% at the median time here. If we look at what Boruta tells us for this cohort, we had 120 diagnostic variables in the beginning. We reduced that set to 56 by data preprocessing, by merging columns and so on. And we kept, I mean, Boruta tells us that only 13 variables out of those have a really significant prediction power. And if we keep only those 13 variables, and if we train again the models, then we only lose 0.7 points of accuracy. And here you can see the SHAP output for this model trained with Boruta for the most important variables for the model. Okay, so it means that are the variables that the model looked at to make his decision. Then we did the same for the ASA cohort. It's much harder to predict. The patients are older and weaker, and so the outcome is more uncertain for our methods. We couldn't get better than 62% accuracy. So it's not very good, but it's not a problem of the models, we think, because we tried many different models. It just means that maybe the data we look at, the variables we look at, don't have much more predictive power than that. So if we do the same using Boruta, we keep only 7 variables for this cohort. And if we train again with only those 7 variables, we lose only 0.2% of accuracy. So once again, this shows that, well, maybe there is not enough prediction power in the variables we look at, because if we keep only 7 variables out of all the variables we have, we don't lose any accuracy. So then we looked at some other source of data that we have for all of these patients in the database. We have diagnostic bone marrow smears, and the cytology, the hematology department scanned all of these smears. And so we have a lot of images that we can use for many different things. We can use it to automatically classify cell types. We can use it to correlate what we see with some genotype, some mutation information. And we can use it to enhance the prediction, the overall survival prediction models that we have using multimodal information. So it's a hierarchical image format. This is the lowest resolution. You can't see anything here, but if you zoom in and zoom in and zoom in, just like on Google Maps, in the end you get to see different cells, so colored in blue here. So one image is 16 gigabytes of data, so it's a lot. It's a lot to handle for algorithms and computer methods that we have. We need to pre-process this also. And the bone marrow smears actually contain a lot of useless regions. Some parts have no cells at all. Some parts here are too dense and the cells are very packed together and we can't see anything. And some parts are blurry because of the way it was scanned. So we need to pre-process a lot and to select small tiles like that one. In the end, we end up with tiles of like 2000 per 2000 bytes. So much smaller than 16 gigabytes. But for one image like that, we have a lot of small tiles like this one in the end that we need to filter. Then once we have those tiles, we use one algorithm that is developed in our team. It's called Kartezio for cell segmentation. So there are a lot of neural networks that do cell segmentation that we could use, but these algorithms need a lot of data to train on, and you need to label this data. So it's a very tedious task, and we are not professionals at this. So we only had the images like a few months ago. And so we did the labeling by ourselves. So the students in our team developed a very nice tool to do the segmentation on like a tablet. And so we annotated only 10 images, and that is the power of this method. It uses something called Cartesian genetic programming to just assemble some very, not basic, but very well-known image processing functions together. So one after the other, and it tunes the parameters so that it can find a mask and a marker. So the mask is how we will segment the cells, and the marker locates the the center of the cell. So if there are like 3 cells very close together, and if we have 3 different markers, that way we can segment differently. I mean, we can precisely do the segmentation even though the cells are packed together. Only 10 annotated images, and it works pretty well, actually. And it's explainable AI, because in the end, what you get is a recipe, is the sequence of functions that you need to apply to your image in order to do that segmentation. And it's very generalizable also. So once we had these segmented cells, for now, we didn't have a lot of time to work on that. The only thing that we started to do is to try some unsupervised classification methods. We just separated a few images in our data set into blast cells and non-blast cells and tried some unsupervised methods to classify those. So to do that, we use what we call an autoencoder. So it's a neural network that takes one image and goes into some lower dimensional space here, that we call a latent space. And from that, tries to reconstruct the image. So the goal of doing that is to extract here the most important features in the images in a low dimensional space. So once you have this space, you can do clustering on it, and you can separate your data into different classes. This is very preliminary work. We don't have very exciting results to show about this yet, but we will have. For now, we only used black and white images, and this is how we could reconstruct it with very basic methods for now. So there is a lot of hope that this will give something in the end. So what we are going to do for now is to keep on working on those images. It's a very valuable source of data, but it's very challenging because, like I said, we have many problems in the preprocessing of those. So we need to improve our algorithms to filter out things that we don't want to look at automatically. And then we will need to implement supervised neural nets for cell classification. For now, we only used unsupervised methods because, well, we need to label all this data if you want to do supervised learning. And then once we have that, we can extract features from the images, and we can try to correlate the images, like I said, with some genotypic information we have from our database. And we can also build multimodal models in order to see if this can improve the overall predictions that we have. Thank you for your attention.